Hello and welcome to our video on protein structure.
So if we think about an analogy for the levels of protein structure, we might like to take the page of a book, for example. So this is just the first page of the very first Harry Potter book. And you know that anytime you read a book, the simplest intelligible symbols are the letters, right? I've got M, R, A, N, D, M, R, S, D, U, R, S, L, E, Y, right? Those are intelligible symbols, and they're the simplest intelligible symbols on the page. Those letters form words, right? We don't read MR, we read Mr. We don't read FOUR, we read for. That's the next level up in structure. Letters form words, and we know to interpret them as words. We don't just read the words in an isolated fashion, right? Syntax is the arrangement of words in a meaningful way. Mr. and Mrs. Dursley of number 4 Privet Drive. We wouldn't say number 4 Mr. and Mrs. of Drive Privet, would we? Syntax is arranging those words in a way that makes sense. We're lining up the words. The next level up is sentences that express a complete thought. We don't just stop at Mr. and Mrs. Dursley of number 4. Mr. and Mrs. Dursley of number 4 Privet Drive were very proud to say that they were perfectly normal. Thank you very much. That's a sentence. It's expressing one complete thought. Then paragraphs expand on a single theme, right? The single theme we've got in the first statement. These are perfectly normal people. They're the last people you'd expect to be involved in anything strange or mysterious because they just didn't hold with such nonsense. We know who the Dursleys are by reading this paragraph made up of sentences. Sentences are meaningful because the words are arranged. The syntax puts them in a way that makes sense. The words themselves make sense, and the letters are the simplest intelligible symbols. Proteins have a similar idea. The letters in protein structures are amino acids. Remember, we've said in earlier chapters that amino acids are proteins. Amino acids make up proteins, and proteins are polymers built from amino acids. There are 20 possible monomers. 11 of them your body makes, 9 of them you have to eat whole, the essential amino acids. But there are 20 total monomers. Remember, each amino acid has a basic structure. There's an amine group on one end, an NH2. There's a carboxylic acid, a COOH, on the other end. And then that carbon in the middle has an H, and it has something else called a side chain. It's those side chains that are different in 20 different ways to make our amino acids. Proteins can hook together like boxcars, right? The amine on one protein hooks up to the carboxylic acid on the other. So here's the old carboxylic acid, here's the old amine. They're hooked up together, and we get an amide. We get a peptide bond. This was an amino acid, this was an amino acid. Right? This is a peptide. This is a chain of 1, 2, 3, 4 amino acids, or a polypeptide chain, multiple peptides. And it's from the carboxylic acid on one, hooking up with the amino acid of the other. These are our letters, single amino acids. This is a word as part of our peptide. The words, the actual order of the amino acids, are what we call the primary structure of a protein, right? Amino acids have an amine on one end, a carboxylic acid on the other end. The R is what makes the letter different from one another, an A versus a B versus a C. And when we hook them up in a specific order, we're making and spelling words, right? CAT is a word that we personally understand as cat, but ATC is not a word. It's the same sort of thing in the primary structures. We string together the proteins, and the primary structure is just the linear sequence of amino acids, and also the location of what are called disulfide bridges. There are a couple of amino acids that contain sulfurs, and those sulfurs form sulfur bridges, disulfide bridges, with other sulfurs and other amino acids. Those are the primary structure, the actual sequence, how we lined up the amino acids. What about the syntax? We've got what's called secondary structure in proteins, and this has to do with the types of amino acids and the side chains that we have. So we've got what are called alpha helixes, or alpha helices actually. An alpha helix is an example of one form of secondary structure. It twists in a similar way to DNA, right? Helices look like coils, and alpha helices are just one type of secondary structure. So if you see a coil in a picture of a protein, you're seeing an alpha helix. There's also what are called beta sheets. We've got a peptide chain, and what it does is it kind of folds in on itself. It makes these sheets. And every time you see these dotted lines, we're seeing hydrogen bonding. Hydrogen bonding between side groups on protein chains is what makes our secondary structure happen. And these are what are called the folding patterns, right? The protein folds. This protein isn't just a long string of thousands of amino acids. It's a string of amino acids that are folded in very specific ways. It can fold into coils. It can fold into sheets. There are some regions that don't have secondary structure at all, and they're a little bit more globular as well. We'll see that in a picture of a protein coming up. Let's go up to the next level. Let's look at the sentences for proteins, and those are the tertiary structure, the way a protein looks. It's the overall structure of the whole protein. This again is insulin, and if we look at a single insulin subunit, we can see helices. That's what these purple and pink coils are. These are alpha helices in the protein, and then this yellow guy over here is a beta sheet, and these white regions in the middle are unstructured or sometimes called globular. Right? This considers the interactions between all secondary structural regions. You notice we've got an alpha helix, and we've got sort of a long string of no structure, but we've got a beta sheet, and we've got no structure, and then we've got alpha helices. They interact, and they have relationships to each other in space. And the tertiary structure says, okay, I've got regions of helices, regions of sheets. This is how they all look relative to each other. It's like looking on a map, right? There's a secondary structure in Paris called the Eiffel Tower. There's a secondary structure in Paris called the Arc de Triomphe. And the tertiary structure is that these things are within a certain distance of each other. There's one more level of protein structure that your book didn't talk about, and that's called quaternary structure. And for that, I'd like to look at the insulin receptor. Remember, this is a receptor protein complex, and what it does is when insulin fits its way in there, your body is programmed to have a specific physiological response. It tells your body to store glucose. And the thing about the insulin receptor is it actually isn't one single protein. There's 4 different protein chains in there. The insulin receptor is a tetramer. It's got 4 different protein subunits, 1, 2, 3, and 4. And those subunits are all part of one big protein complex. Right? Here's a protein subunit with its primary, secondary, and tertiary structure. Here's another one. Here's a third one. Here's a fourth one. So there are 4 sentences in this overall paragraph, basically. So I want you to know those different levels of structure, right? Primary structure is the actual string of amino acids, what their identity is. Secondary structure is those regions of alpha helices and beta sheets. Tertiary is the relationship of the secondary parts to each other. And then quaternary is the next level up. Sometimes you'll hear this called a protein complex as well. So a protein complex implies that there's quaternary structure.